Our speaker today, and this is our last science seminar for the semester, by the way, right? Nice to see you all here, and hope you've had a good time. If you have ideas for seminar speakers for next semester, we still have a few open slots left. So if your dad, or your mom, or your brother, or your aunt, or a cousin, you know somebody who would make a very good speaker, let us know about that, and we'll try to get them into the series for next semester, right? Or if you guys have something you want to talk about, right, let us know that as well. But today's speaker is Todd P. Prim. P stands for Paul, or
Nobody knows.
Secret. Yeah. And he is a biochemist, PhD from Baylor College of Medicine. And he is now at Sam Houston University. He's been there for about 10 years ago. He was chair of the department there for a while, but then they promoted him to become director of PACE, which is professional and something else there, right down there. Yep. So he'll tell you all about that. He's going to talk to us today about antibiotics and the microbiome. So let's give him a warm welcome.
So, thanks a lot for the invitation. It's nice to come up here. I've always wanted to visit LeTourneau. I've known about it for a long time. Of course, it's pretty well known among Christian circles. So I've always known about it and always wanted to visit. So I'm glad I get to hang out with some of you guys later and maybe do the tour and whatever. So yeah, and I've known, mainly I got this because I've known Greg, well, Dr. Frederick, for a really long time. So that's kind of why I'm here. So I'm sure you all know what antibiotics are. You may not know what the microbiome is, but we will shortly. So I pulled this title out, which probably most of you don't know. The faculty do. Most of you don't, because you're children. So, but there was a great classic movie with that title. Okay. But another subtitle I have is based on the model organism that I use, which might be a surprise to you to model human medicine, but I use a fish. So the alternate title is Fishing for Answers. So a little preview of what we're going to do. In case you lose me in the middle and doze off or something, you can catch up. Okay. Talk a little bit of background. Okay. And a little bit about antibiotics. You probably know most of the good. I'll explain some of the negative possibilities, and then kind of focus in on our key questions that we work on in my lab. Okay. And then the model system we use to analyze that. And then talk about finally a little bit of cool data. Okay. I tried to not do too much of that because I know most of you are engineers. You're not a micro nerd like me. So, but I put it in there. Okay. I have to. And then a little discussion at the end. Okay. And hopefully we'll do a little Q&A and fun stuff. Okay. So, first thing is, so a technique I use in some of my classes is something I invented. Basically using sign language to see what everybody's thinking. The way this works is I show you a question, multiple choice, I give you about 15 seconds or so to analyze it, come up with your answer, and then everybody holds up their sign all at once. Okay? Basically it's cheap clickers. Okay? Because I'm cheap. Because I work at a state university and we're cheap. So, so basically you just make one of these signs here. Alright? So let's try it. So the first question is, of course, what is a microbiome? If you don't know the answer, it's fine. But what is a microbiome? Okay? So here's your choices. Look at this for a second. Okay. Now, show me your sign, everybody. Let me see your sign. Okay. So we have a bit of diversity, although just normal. That's cool. I like using this in class because I can just really quickly survey everybody. If everybody answers it once, I get a more authentic feel of what everybody's thinking. Because if we Otherwise, everybody just looks at Smarty Pants at the front row and just answers what she answers. They don't really think about it, and that doesn't do me any good. I want to know what people are thinking while I'm interacting with them in the classroom. So those of you who picked C, yes, you're correct. Okay. So a biome is another word for an ecosystem, although a biome actually refers to the organisms in an ecosystem. Okay. Ecosystem is both abiotic and biotic, but anyway, a microbiome is a biome where you're looking at microorganisms. Okay. And in my context, really the microbiome is looking at all the organisms that are on an animal. So you're in a very elaborate ecosystem, basically, to me. I see you as a giant walking island. Okay.
No man.
Microbiologists see a lot of interesting things. So. And this has been explored a bit with, how many of you have heard of the Human Genome Project? Good. How many of you have heard of the Human Microbiome Project? One. Okay. She gets an A. So, Human Microbiome Project is a very ambitious project to basically map the diversity that's on humans at the microbial level. What is there? And then you can start to ask what's it doing, what's happening. So, an extensive project. I can't go through all of it, but basically, how can you answer a question like that? It's really challenging. Humans have as many as 100 trillion different microbes on them. There could be, there's probably as many as 1,000 different species in your gut. That's an exceptionally elaborate ecosystem. So what's allowed us to actually now start actually answering a question like this, because before you could only dream about answering it, really is DNA sequencing technology and the way that it has gotten so drastically cheap and rapid. I'm assuming you guys know what Moore's Law is, since you're all engineers, or most of you are engineers. Right? But notice DNA sequencing technology has made Moore's Law look like, a turtle. So basically the Human Genome Project costs several $1 billion to sequence the genome of one person. Now we can sequence the genome of one person for about $12,000. The cost difference there is like me selling you a brand new car for a nickel. Okay. So, and we can sequence massively in parallel. One run on a DNA sequencer now can give you 400 million DNA sequences. So now we can ask the question, what's all there? And actually measure it. And it's not even a crazy thing to ask. Okay? So that's really cool. How do we do that in microbiology? Well, a key tool we have is something called a 16S profile. Basically this is, you take a snapshot of an ecosystem and you say, what are all the microbes there? Everybody. And you do it in a way where you don't have to grow them in a lab. Because not everything grows in a lab. In every condition in a lab, everything doesn't grow in one condition. So this is a culture-independent snapshot of everybody that's there. It's like a census of the entire country. Poof, one snapshot. Okay. How would we do something like that? Well, you extract DNA out of a bacteria, and you sequence a fragment of a particular gene called the 16S gene. Okay. Why do we pick that gene? Because it's in all bacteria. Because it makes part of the ribosome, which of course, all bacteria have to make proteins, so they all have to have a ribosome, so they all have this gene. So what you do is you use PCR to amplify up a fragment of that gene. Then you do your now super fast, cheap DNA sequencing. Then you take all the sequences that are similar to each other. Now you have them all on the computer, and you bin them. Then you take a representative of each bin, you compare it to the database, and find out who is that. Okay. So a typical run for us might contain 80,000 DNA sequences, and we can match all those up with not too much. Spawn an entire field called bioinformatics, which if you're into computers is pretty cool stuff. We'll talk about that later if you want. So basically, this is a key technique. I just wanted to blow this one up, so when I said it, it didn't sound like Chinese. Okay? So what have we found in the Human Microbiome Project? I can't tell you everything we found because it's pretty extensive. But basically, they took 242 volunteers, okay, who are close to your age. Okay. They were extensively screened so they're healthy. In other words, there was like 70 or 80 conditions they couldn't have to be excluded. So these are normal, healthy people. So a bunch were in Houston because Baylor College of Medicine was part of it, but many were in Seattle as well, and some in, I think, St. Louis. But anyway, 242 people, and they sampled up to 18 body sites. all of them. Okay? Some people were sampled once, some twice, some sadistic people even 3 times. Okay? This is all volunteer. Okay? And then they sequenced away. And what's on people? A lot of stuff. Okay? As you might expect. Okay? So it turns out that the bacteria on your body outnumber your cells by about 5 to 1. So if you did a population census, you're more microbes than you are a person. But more impressively, really we're talking about the interaction between gene products, your gene products and the gene products of bacteria, really. Okay, well, humans have about 25,000 genes. All the microbes in your community together have several million unique genes, so they outnumber you thousands to 1 as far as genetic contributions. So the microbiome on a person contributes to digestion. They make a number of unique vitamins that you can't make. They process a lot of carbohydrates and things that you can't process. They also contribute to, in fact, 2 people can eat exactly the same food and get different calories based on your microbiome. Okay. They also explain some things. It's my microbiome. No, I think it's the 5 donuts. But anyway, it also dramatically affects our immune system, training of our immune system, whether or not possibly you get allergies or something later, as well as a lot of chronic diseases are related to it. It could possibly influence behavior, development, many, many, many other things. Microbiomes, we're just now starting to unlock this box. Okay? So this is really a watershed study, fascinating. Okay? So you're an elaborate symbiotic system, and many microbiologists now refer to your microbiome as your other organ system, all that it does for you. Okay? Can humans live without a microbiome? Yes, well, probably. There's one example, possibly, David the Bubble Boy, which again you probably don't know about because you're too young. I remember seeing him on TV when I was a kid. So, another question here for you. What is a broad spectrum antibiotic? So look at it for a second. Pick your answer. Are you ready? Okay, show me your answer. Read the words to me again. Looks like some spread around, okay? A would be close, but really the best answer here is C. Okay? There's no antibiotic that affects all bacteria. Pretty much if you put any phrase next to the word all bacteria, it's not true. Because they're so diverse. I mean, maybe all bacteria have DNA, but beyond that, it's pretty much a toss-up. Okay? But broad spectrum antibiotic affects many different species of Genera. A narrow spectrum affects less. Okay? So one would predict a broad spectrum antibiotic would affect your community more, maybe. But anyway, so antibiotics. Antibiotics are great tools, right? They're a very powerful part of modern medicine. Very potent. Okay? However, there are downsides. Nothing is free. Right? Hopefully you know that already. When people say this is free, they don't really mean it's free. Right? It means just someone else is paying for it. There's no free. Okay? So I just spoiled the presidential debates, but there's no free. Okay, so question. If you're looking at your body, right, it's basically an elaborate ecosystem covered with enormous numbers of microbes, right? And then you have one bad microbe coming, a pathogen, right? So our mental model here is an elaborate rainforest, okay, and fire ants, okay? There's your bad guy, okay? How are you going to get rid of that, okay? Well, if it's bacteria, you might take an antibiotic, right? The antibiotic does not just kill that one thing, right? Depending on which antibiotic it is. So basically, in some ways, it's somewhat similar to doing this, okay? To get rid of these guys, okay? And as you know, when the forest returns, it can return in a number of different ways. This is Yellowstone post the giant fire in the 80s. Okay. So, collateral damage is basically what I'm interested in. Okay. When you take this antibiotic, you're affecting an entire ecosystem. So lots of people look at that pathogen and how the antibiotic interacts with that, and that's important and it's good. I step back and basically do microbial ecology and go, what are you doing to the whole system? How does the system respond? How does the system come back? Okay, and might this explain a number of issues that humans have when they take antibiotics, and animals as well. Okay, one of the most common problems after taking an antibiotic is diarrhea. That's just a clear phenotypic marker of, hey, something's wrong in your gut, right? And depending on the antibiotic and the human population, sometimes the rates are 2 or 3%, or sometimes they're as high as 30% of the patients getting an antibiotic-associated diarrhea, which sometimes progresses to enterocolitis, which can be quite negative. So, what are you doing? You're messing up the ecosystem. So that's an interesting question to us. Okay? So, here's where we are. So look, we're like almost halfway down. All right. So again, lots of people study this classic question, right? You have a bad organism, you take an antibiotic and you kill it off, that's cool. It is interesting, but we're more looking at ecosystem. How does it affect your microbiome? It's a tougher question, but that's the interesting thing in science, the tough questions, right? What happens to your community composition? What happens to your community dynamics? What happens to your community function? And then what happens to the human related to that community, since we're so closely related, connected to that community? Okay. Here's actually, you don't have to read all this, it's just a placeholder. This is an interesting study by a really big guy in the microbiome field, David Relman, looking at humans who took the antibiotic ciprofloxacin. You've heard it called Cipro. Okay. Humans who took Cipro, and he tracked them for like 8 months. Okay. This is poop samples, so he's looking at the microbiome. If you're a microbiologist, you get used to poop because that's just it's part of the job. Okay. It's a poopy job. So, and what he noticed, pretty interestingly, is it did change the composition in the gut microbiome of those people. Okay. About 2 or 3 days after they started taking the antibiotic, and about a week later, the composition shifted close back to what it used to be, but not all the way back. So there was some semi-permanent change. Very interesting. Now this study is fascinating, it's really cool, but there's only 3 people in it. Because working with people is expensive, it's challenging, and you can't manipulate people and control their diet and all that stuff. At least the FDA frowns on that. Okay, right? It's unethical, right? You can't control everything about it. Studies with humans are really, really, really messy, okay? So, what we need is a model organism. A model organism is an organism you can take in the lab and you can control everything, exactly so, and therefore you can get more causative data. And then go back to the humans and try to see what's going on. And that's what we do in biology all the time. That's the foundation of biomedical science, in fact. So, one more question for you here. What do you think is the most common model organism used in the world for biomedical science? Take a look. All right, give me a sign. Let me see. We have a variety of answers here. Most everybody is picking our little fuzzy mouse, which is correct. About 92% of all biomedical research is done with mice. There is some research with the other ones I listed here, but very, very common. And the genetic things that we've been able to do with mice to understand human disease are perfectly astounding. In fact, it's now quite possible to take a mouse, which has no immune system, and take bone marrow from a patient, put it in, give the mouse basically that patient's exact immune system, and then transfer tissue from that patient into the mouse and then model a disease with that. That's the science that we have for biomedical research now, which is astounding. But I'm not using mice, okay? I needed something different, which I'm hoping we'll get to here. Probably not, okay? But it's pretty cool, and I use actually a fish model, okay? Not this fish, this fish, okay? Which you may or may not have heard of, probably not. This is the scientific name. The common name is called the western mosquito fish. They're about yay big or so. So they're cheap, they're easy. I can have 70 individuals in a tank this big, okay? And they're also a vertebrate, so they have vertebrate-acquired immunity, so their immune system is complicated in the same way ours is and different from, say, an insect or something, okay? And what I look at is their microbiome, and I look in two locations. One, I look in the gut, but I also look at the skin. And here's the cool thing about this model organism. I'm really interested in what we call mucosal microbiomes. So mucosal surfaces in humans are obviously your gut, mouth, lungs, right, reproductive tract, etc. Not the skin. Okay? But in fish, their skin is actually mucosal immunity. They have mucus all over their body. If you've been fishing, you've held fish and they're kind of slimy, okay? So they're covered with snot just like your lungs are, right? Well, we call it mucus because snot is not really scientific, but anyway, you get the point. So what's the beauty there? The beauty is I can easily sample their mucus. Right? I don't have to do a scope or something crazy like that with a mouse. I can just swab off some of their mucus layer and the microbiome in it. Or I can just change what's in the tank water and immediately expose the fish to different pH, different chemicals, anything I want, I can rapidly manipulate the conditions. And I know exactly what the conditions are. And then I can sample repeatedly. And that sampling is almost always non-lethal. So they're a handy-dandy system for mucosal microbiome research. Okay. So far, I think I'm the only one using this one, so it's not really catching on, but I'm not always fashionable, so my kids will tell you. So the first student, so one of the themes I wanted to talk about here is undergraduates involved in research, which is super important to me. That's my favorite part of the job. This is the first student who established this fish model in our lab. His name's Robert Fultz. He's now graduated. Now he's actually in a PhD program at Baylor College of Medicine. Here he is receiving an award at a recent meeting that we had. So Robert started it off. So what antibiotic are we using as a model for antibiotics? We're using this antibiotic called rifampicin, or rifampin. Okay. Why do we pick this one? Well, number one, it's a broad-spectrum antibiotic, which is what we wanted to analyze first. Number two, it's cheap. We have already established I'm cheap. Okay. But number three, most importantly scientifically for the study, is rifampicin is easily distributed among tissues evenly. In humans, it's often used to treat meningitis. Most antibiotics can't get into the nervous system, so this is used to treat meningitis. So for that reason, if I expose fish to it in the water, one, it's not lethal to our fish, but two, it's going to distribute around really quickly and easily. So that's mainly why we picked it. It's also bright red, so it's easy to see that it's in the water. I would say that it kind of looks like Chablis or something like that, but you know, we don't drink here. So anyway, if you did, it was kind of like wine. So, all right. So, what do we think is going to happen? What we think is going to happen is basically looking at other studies. We made some straightforward predictions. If you take the fish and you expose them to the antibiotic for a little bit, and in this case a week, you expect the total numbers of bacteria would shoot down initially as you're killing some off, but they would adapt and come back in return. The same thing to the diversity of the community. Diversity means how many different members in the community. So if you started out with 1,000 species, maybe it would shoot down to, 500 or 600, but then come back. And then the percent of bacteria that are resistant to the antibiotics initially should be low. You'll select for some resistance, and then that should go away again. These are our straightforward guesses that we made to start off with. Well, they're hypotheses because we're scientists, they're not guesses. So what do we actually see going on? Okay. Well, basically our study design. So we collect our fish from one site. It's this pristine lake. Really, it's a pond, but they call it a lake. Basically, it's undisturbed, very natural, nice habitat we can collect from there. And then we adapt them to the lab conditions briefly. And then in this case, again, we just put them in the antibiotic for 1 week. That's a fairly typical exposure time for humans. It depends on, of course, what infection they have. But 1 week is fairly typical for antibiotics. Okay. And we did it at a fairly high dose. Okay. And then we took the fish and did a water change. And then we just observed for a couple of weeks after that, how did they recover the microbiome. How do you sample the microbiome? Again, it's really easy because it's external. You just capture your fish, okay? and you put them in a buffer that has a little bit of salt and a little bit of detergent, so it disperses this mucus off their skin, and you basically just give them a little spin. Okay? And then it's all approved by IACUC. Okay. If you do research with vertebrates, you must go, there's certain rules about that, of course, and everything has to be approved by what's called an IACUC, the Institutional Animal Care and Use Committee. It's all approved. And like I said, usually, unless the fish are really stressed before you do this, they almost always survive. The beauty of this is you just give them a spin, and now their microbiome is suspended in this solution. Of course, once you have it in a liquid, you can do a lot of analysis. The analysis that we do include classic plating, to get the total number of bacteria, but also to get the percent that's resistant to the antibiotic. If you just have the antibiotic embedded in the media, they have to be resistant to grow. We also looked at a biochemical profile analysis. Functionally, what biochemical activities does that community possess? So it's a functional look. Okay. These are some of the strips that we use. We use a couple of different test strips that link every biochemical reaction to a certain color change or something. Or we can look at the antibiotic susceptibility profile of the community. Or finally, we can do our 16S profile that I mentioned before and take a snapshot of who all is there. Okay. So, finally, what are we actually seeing? So this is the culture analysis, counting colonies on plates. In black is the total number of bacteria colonies that we get. Okay, and you can see this is a time period across the bottom. This is the 1 week of antibiotic exposure, and here's the recovery. So, somewhat as we predicted, the number shoots down quite rapidly as you're killing off everything, or almost everything that's sensitive to the antibiotic. And then it bounces back, actually, really rapidly. So Yellowstone is still recovering from the fire in the 80s. My community came back in a day. So microbes move at light speed. Okay. And in fact, kind of interestingly, the stable number was a bit higher than where it started. So the community kind of overgrew a bit. Ecologists sometimes notice this when you disturb a community dramatically. The niches, there's a whole bunch of niches that are not occupied, and you get a temporary overgrowth phenomenon. Okay. Although that eventually kind of persists a bit. Kind of returns to fairly close to the same number. Do you have a question?
Is it the same dispersion of bacteria that came back rapidly, or was it a certain type?
Looking at the colony, what we call morphotype, the appearance of the colonies, there's definitely some changes. It's difficult to be clear exactly what those changes mean, okay? But there were definitely some changes. 16S profile is a much better way to look at that question, so it's a good question. I'll save the real answer for that in a minute. And the red line is the colonies that are antibiotic resistant. So notice, there's very few, but it's below detection limit. These bacteria that are normally in the pond are normally adapted to the lab. It's less than 10 bacteria per fish are resistant to this antibiotic already. And notice, when they come back, they're basically 90% resistant already. But here's the curious thing. We expected that to peter off once you remove the drug. But actually, it stayed fairly constant at 20% resistant. I don't know why that persisted in the population. That's kind of unusual. We're still trying to figure out what's driving that. Okay. But that's curious. Okay, what about the biochem? I know you can't read this table. You don't have to read the table. These are the biochemical activities that we analyzed in the communities. Here is 3 different fish before antibiotic treatment. Okay. And here's 3 fish during the 3rd day of antibiotic treatment. So we know that the total number of bacteria has already returned. Okay, but you shifted, possibly shifted what's there. And what I'm showing here is biochem activities that were lost are in red, and ones that were gained are in yellow. So basically this suggests there's a very strong, about a 1/3 of the functional capacity that we analyzed changed. That suggests the community has shifted quite dramatically in what it can do. Okay. Here's actually the antibiotic resistance. I know, again, it's a big table. I know you don't have to notice. You don't have to read everything. One thing, the first thing that you obviously notice is you go from somewhat sensitive to fully resistant to rifampicin. That's not a shocker. But we also noticed a couple of other antibiotics it became fully resistant to. And these antibiotics are pretty much unrelated to the function of rifampicin. So why that appeared, I really don't know. It's a community change, so it's a complicated question, but a surprising result, which is all the science finds. So let's get to the fun stuff, the 16S profile. What's there? This is a pie chart showing you what's present on the fish before treatment. So this is 4 different fish, were all analyzed. Okay, we just looked at the skin microbiome, what's there. Okay. And what you do is percent normalized abundance. You know, like looking at the population of, you know, Lanterno, 22% of the students from Texas and 5% from China and blah, 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 right? This is the same thing except bacteria. And it's at the level of genus. The piece of DNA that we're sequencing is not long enough to tell down to the species level, so we can tell genus level. And these are some of the bacterial genera that you probably don't recognize, but that's okay. So here's the interesting thing. The community on the skin of the fish is very, very diverse, but it's very, very uneven. What do I mean by that? There's about 400 different organisms there. But notice the first 5 are over half of what's there. So those 5 are half. The other 395 are half. So it's very, very uneven. One organism is 21%, and there's a whole pile of organisms that are 0.0001% or less. So it's extremely uneven. This is not too common. This is actually fairly common to see in microbiomes. There's some organisms that are really stable and kind of establish a niche and take over, and they kind of defend that area. And the top 7, which I colored here, are about 70% of what's there. So it is a highly dominated ecosystem. That's not too surprising, actually. It's also quite stable because these top 5 were the same top 5 in all 4 fish. They might be moved around in percentile a little bit, but they were always the top 5. So it's pretty consistent among fish. Probably not a surprise because they're all in the water together and they're kind of, their skin microbiomes are fairly homogenized. Okay, so what happens when you drug them? That's the big question, right? Okay, here's a chart across time. The pie chart you just saw has been converted to here, pre-treatment. Okay, and then here's across time different samples that we're taking during the drug treatment of 1 week. So what this is showing you here with these colors is that the abundance of each different organism is varying quite dramatically. In fact, these original top members that are about 70% of what's there, by a couple of days, they're only about 1%. So they've been completely smashed. Okay? But here's one surprising finding we found is that one organism, which started out as 0.002% of everything there, jumped up to be 95%. This one guy just completely took over. It's called Leroyes. Okay? But that's not stable. And then he declines way down. And another group of organisms here, which start out as a tiny fraction, kind of take over. So you shift from one dominated microbiome to another dominated microbiome. Very dramatic community composition shift. And an intermediate stage is this one guy taking over. You have preliminary evidence with another antibiotic that the same thing happens. It's a different organism, but that one taking, so that might actually be a general phenomenon of antibiotics. I'm not sure yet. 2 is not enough to say it's general, but it could be. So it's kind of a fascinating and dramatic shift. So the microbiome does restabilize, but it restabilizes to something different. Okay? These are organisms that seem to gain in the end, okay, for reasons that are as yet unclear. Okay? So this is what's called this is a type of statistical analysis called a principal components analysis. This is where you take different facets of your component, okay, and you graph them out. Basically, each community, each sample point from the fish, that one community, is each represented by 1 dot. Okay. So the 4 fish before antibiotic treatment are here. Notice they're pretty similar to each other, and they're clustered up over here. Then when they're exposed to the antibiotic, that's these yellow dots. So notice you're blasting the community to be very, very different, but you're also spreading the fish out from each other to more divergent. But here's the cool thing, when it recovers, they cluster back together a bit again, these green dots, but they do not return to the original community composition. They're statistically different. At least 2 weeks it doesn't return. Now, this is a broad spectrum antibiotic at a high dose. So in that case, it's somewhat of a worst case scenario. But it does show a dramatic community change which does not return back. There are some studies in humans where, again, they mostly come back. Humans seem to be a little more stable in this, maybe, but definitely can change the community quite a bit. Okay? So you get a rebound, yes. This was totally wrong, okay, because it went to a very different community. And this was kind of right-ish, okay, but we had some persistence of resistance for reasons that we don't yet understand. So, success in science is not having a correct hypothesis. Success in science is correctly testing your hypothesis, okay? And in fact, scientists usually discard your hypotheses because usually we're wrong, because nature is dramatically more fascinating and complicated than we think about. So it's okay to be wrong, okay? Just actually know that you're wrong or not, okay? All right, so for those of you who have lost and you kind of lost, here's how you can catch up, okay? So here's the fish before antibiotic. It's dominated by 1, 2, 3, 4, 5, mostly these guys, and extremely rare there's a resistant guy in here, okay? You expose them to the antibiotic, and you shift to a new community, which is now dominated by A, B, C, D, E, okay? So the structure is somewhat similar, a dominated community, but who's dominating is not quite different. And now, most of the guys are resistant. Okay? So that's my simplified catch-up, okay? Okay, that's kind of fascinating and interesting, but really the real question is, okay, so what? Right? Okay, you changed the microbial community. Yippee. Right? No, the real question is, okay, what does that do to the host, the human or the fish? What does it do to them? Does it have any effect on them at all? If you change the microbial community and the fish is perfectly fine, okay, that's interesting, but really, I mean, what's the big deal, right? So we really wanted to know host effect. And this has been explored much, much less than how the community has changed. And we think we had a model where we could change it. And this is actually started off, it's continued, but it started off as a summer project from a research program that we had at the university. And these students were involved in it, Jeanette and Dana and John. Okay, they all did this in the summer. They were funded to do this research, and then they continued it on, and now we've actually just last week published this study. So the effect on the fish, all right? So this study is slightly different. We take the fish and we expose them to antibiotics for 3 days. That's long enough to drastically change the community on the fish. We know that. Okay. And then we let them recover. But while they're recovering, we're analyzing some capacity about the fish, some phenotype. What do we analyze? Several different things. First thing we analyzed was how well can they resist an infection? Okay. One part of the microbiome that's proposed most commonly is what we call competitive exclusion. Basically, appropriate for Christmas time, there's no room at the end, okay? So, if all the niches in you are full and you eat salmonella, it has no place to go, so it just passes through. That's called competitive exclusion, okay? Our way to analyze that is expose them to a known fish pathogen. Okay? But we also, as a control, wanted to expose them to just lots and lots of microbes, basically simulate a really dirty environment. Okay? We use two common sources of complicated, large numbers of microbes, soil and poop. Okay? And, I told you, microbiologists love poop. You just can't get away from it. So, another thing we wanted to examine is a physiological stress that's really common to fish, and that's changing the salt in the water. And finally, we wanted to look at any nutritional effect that might happen. Okay. Feed them and look at them gaining weight over time. So, first thing, okay, this is what's called a survival curve. How you read this is it starts off Each group is a line. You have a group of animals, and they're all 100% alive when you start. And over time, as members of that group die, your percentage survival drops off. It kind of looks like a stair step. So what's going on here in black is fish that were not exposed to the antibiotic, and then they were exposed to this pathogen. Okay, same dose. A different group was exposed to the antibiotic for 3 days, then they were rested for 1 day so the antibiotic gets out of their system. The antibiotic's not directly affecting the experiment. Then we expose them to exactly the same dose of the pathogen. And what we noticed is, and you can see, they die a little bit faster. Not dramatically, but a little bit. In other words, they're more susceptible to the pathogen when their microbiome has been changed. It's a verification of competitive exclusion, like many people have proposed before. So that's really kind of interesting. Note, after 3 days, the total number of microbes should be about the same. It's just different who's there. Okay. So that was an interesting finding. Not too surprising, confirmed what a lot of people thought, but interesting. Okay. When we follow that up with our study looking at the soil and poop, okay, I gotta love nerd humor. So, stool sample, you get it? Stool? In microbiology, that just lays them out. So anyway, when we did this experiment, I'm not showing you the data because basically there was no change between the two groups. So when you just expose them to lots of microbes, they're about the same, but when you give them a specific pathogen, they're less protected. That's interesting. Okay. The next experiment was osmotic pressure. The way we did this is the fish typically grow in a pretty low osmotic strength. It's called artificial pond water. Okay. So after the antibiotic exposure, or the control without the antibiotic exposure, we shifted them to saline that was about half the strength of seawater, which is pretty stressful for them. And notice that the control fish, none of them died until about a day, and then about 40% of that group died. But the ones that were exposed to the antibiotic started dying earlier, and 88% of that group died. So they're dramatically more sensitive to osmotic pressure. For some reason, they're less able to balance salt in their body. Nobody's ever noticed this activity before, so I'm not quite sure what to make of it. Dominantly with fish, the gills are what doing a lot of this osmotic balancing. So we're going to return and actually study the microbiome specifically on the gills, as well as try to look at ion transport and things like that. Take a little more time to figure out why. Of course, the why is really interesting, but this is a fascinating finding. I don't know what to make of it yet. Okay. Finally, the nutrition study. Okay. What's going on here is you have fish that are not drugged or they are drugged. Then they're actually individualized into cups. And in every cup, the exact same mass of food is added every day. And then once a week, you weigh each fish. We only weigh them once a week, so we're not stressing them out too much. So you do that 4 times for an entire month. Okay? And this is showing you individual fish after either 1 week of being in their individual cup compared to 3 weeks. Okay? And since these are hard to see, I'm going to yank this fatty out here. Okay? This is a controlled fish who did gain a little bit of weight. I'm going to pull her out because she's huge. Okay? And blow up the graph a little bit. Okay? So what I've colorized here is C means control fish. If it's colored black, they did gain weight, as you would expect over that period. Blue is the one who did not gain weight. There's always one weirdo in every group. You know what I'm saying, right? Your little brother or something, right? You know, there's one in every group, okay? That's animal work. They just don't always behave. So, but here's interesting. Fish that were treated with the antibiotic, E for exposure. In red are ones that lost weight. Okay? And in green are ones that gained weight. So more of them lost than gained. That was really interesting. But we really wanted to confirm this because being individualized like that is not exactly normal for these fish. They're fairly social. So we wanted to repeat it with group experiments where we put fish together in groups, small groups, in a tub. Okay, much more normal for them. When you do that, we don't mark them so you don't know which fish is which. You can't individually, all you can do is get an average mass per fish over time. Okay. is the averages of this group, but I want to go to the group data. So here's some of the group data. Here's a table of the group data. 2 trials. The first trial, the control fish on average gained 23% of their body mass in a month of eating. They can gain weight fast. The antibiotic fish on average lost 20%. The second trial, you can see they gained better, and these lost less, but still gaining versus losing. So, something's wrong. I don't know if it's that they're not absorbing nutrients well. I know it's not appetite, because we measure how much they eat, and that's about the same. That averages out after a day or 2. But I don't know if it's being able to take up the nutrients, or I don't know if it's the transit time through the gut has been changed, or maybe their gut's inflamed a little and they can't absorb nutrients. There could be a lot of different possibilities. So we're hacking away at those one by one. We've developed a way to look at inflammation in the gut now, by neutrophils going in. We've also developed a way to measure transit time to the fish. You feed them these little tiny fluorescent plastic beads and wait for them to basically come out in the water, and you can measure transit time to the fish. So we're working on trying to figure out why, but this is really interesting. It's linked to, sometimes when young children take broad-spectrum antibiotics, they have failure to thrive. They don't gain weight well. So this could be linked to that. Might be able to figure out something by analyzing what's going on here. Okay, so what's up? Basically, you have your fish with their normal, working, functional microbiome, kind of co-evolve together, if you will, and then you get this ugly thing when you cause a massive disruption. How long does this take to come back? Interesting, I'm not sure. We need to do studies where we examine the fish longer. Okay? And how negative is this? Again, I'm not sure. A lot more study needs to be done, but this is really a fascinating area, I think, to examine. Okay? I may explain lots and lots of things. Okay? So in the future, what are we going to try to focus on? One, again, get behind the hook. We have some clear negative host effects. We want to determine why are they negative. Are you stimulating lots of inflammation? You messed up the immune system? Or you changed transporters in the fish? Or just the bacteria that are there actually damaging the... What's going on? I don't know. 2, we want to look at the community members in the fish community. How do they interact with each other? Right? Can one bacteria functionally replace a different bacteria? Are there some bacteria that are antagonistic towards each other? What's kind of the community network, if you will, going on there? Okay? Because maybe the new community is far less functional because its community network is mostly antagonistic compared to the normal community that's there. I don't know. Next one, we want to, of course, run much more antibiotics. We've already run the antibiotic tetracycline, which is also a broad-spectrum antibiotic. We're collecting all the data on that. But we also want to run several narrow-spectrum antibiotics. We tried a couple, and they unfortunately kill the fish even at pretty low doses, so we have to, because these antibiotics are developed for people, so we have to migrate until we find one that won't hurt them. And then finally, kind of a big application we're excited about is probiotics. Probiotics means treating a disease with live organisms. Okay? We want to take probiotics from a, hey, take yogurt, it'll make you better vague stage to this specific organism can help improve this specific condition. Okay? We want to get to that level. Okay? So, maybe eventually something like that. I don't know. Where, hey, use this thing and you're doing better. The beauty of probiotics is it doesn't spawn antibiotic resistance, which you know is a big problem. So there could be a lot of potential there. Okay? A few other students I should mention. So after Robert, Sonia took over, who's now in grad school at UNT actually, not too far from here. Okay, and then this batch came over. Okay, Annie's in med school, Sarah's applying to med school, and these two, Sonia and Nubia, are actually in nursing school now. Okay, and then finally, my current batch, two actually grad students in our, we have a master's degree at Sam Houston in biology. Talk to me about that later if you're interested. Chelsea and Jeanette, who did her undergraduate work, you remember, with me as well. And then these 3 yokels here. Okay, actually John is going to apply to vet school. Brandon's already gone to med school, and Corbett is still trying to figure out his life. You know what that's like, right? So. Oh, and I forgot, Oscar. Oscar's in pharmacy school at A&M now. So, and these are our collaborators at Baylor College of Medicine who did the DNA sequencing and some of the DNA sequence analysis. Imbriette, a very talented grad student there who's now graduated and has her PhD. And my good friend Joe Petrosino. We actually went to graduate school together when you were babies. Okay, so he has a really impressive center for microbiome research there. So, if you guys have any questions about this, I can be happy to take those questions. I also have to thank my fish, of course. So, any questions related to this research?
Let's thank our speaker. Okay, questions on your microbiome?
Fish? That's a great question. In the wild, it's pretty hard to tell. In the lab, they can last quite a while, probably 4 or 5 years if you keep them away from predators and stuff like that. They're what most people call a minnow. So obviously, they're basically what's for dinner. Everybody else is trying to eat them. So in the wild, they don't last super long.
I'm just curious because in the food industry, a lot of producers of beef and also poultry routinely give antibiotics, and their argument is that the animals increase weight more quickly when they do give those antibiotics. So there seems to be a lack of correlation between, is it dose related or proportion
Yes.
related?
Good question. So, yes, growth promoters in agriculture. Those are very low dose antibiotics. It's much lower than the therapeutic dose. In our experiment, this is a pretty high dose. So it's probably a dosage thing. We're actually trying to do some low dose studies with our fish to see if we can replicate improving growth as well and start figuring that out. Interesting thing is they're not entirely sure why the animals grow faster and gain more weight. Clearly they do. There's no doubt that they do. It works. Exactly how it works, we're not sure. I doubt that there's probably some changes in the microbiome. Exactly what's going on, I don't know. Some people think maybe it's short-chain fatty acid production by the bacteria, degrading carbohydrates better. We really don't know.
What is the total time it takes to run from getting the fish from the pond to finishing the 16S scene and showing the graph before us now? How long did that?
Good question.
Entire process
A bit of time. So capturing the fish is pretty easy. We just go out. We have little nets. Just capture them, dip nets. We need to acclimate them to the lab about a week so everything's stable. Okay. Then the experiments we run are typically 2-3 weeks or something like that. We ship off our samples for DNA sequencing. How long it waits depends on just what they're doing. But once they actually, the sequencing itself, we use a platform called Illumina technology. It's actually about a 2-week run to get the DNA sequence because it generates 4 million DNA sequences at once. So we're like a tiny fraction of what they're actually running at that time. And once you get the DNA, so that's pretty fast. The slower part is analyzing. The Excel sheets I work with have thousands of, it's a lot of data and you have to sit there and analyze. It's a lot of analysis. So that's kind of the slow point. That's also the fun. So one question I get a lot, so of course I'm at a secular university, right? And I'm a scientist, so of course, you can probably guess one question I get a lot is, okay, how can you be a scientist and a follower of Jesus, right? A Christian. As if those two are in juxtaposition in some way. Okay? I get that question fairly a lot, actually. The answer to that's fairly complicated, but one way I answer that question is, okay, in science, if you have a limited amount of data, okay, right, you know certain things, you don't ever know everything, you know certain things, okay, then you can take those certain things and you can extend from what you know and predict into what you don't know. We call that extrapolation. Right? It's extremely well established in mathematics and science to do that commonly. In fact, we do that for most everything because you can't know everything. So extrapolation is a firmly established scientific principle. But I mean, in many instances, we don't know everything about the universe or human nature or things like that, but what we do know actually a fairly good amount, and in fact God has revealed to us in the Bible quite a bit about it, enough about himself so that we can extrapolate from what we do know into what we don't know. There's lots of questions where my answer is I don't know, but I do know that God's in control. And I do know that he cares about us if he's willing to become a man and suffer and die for me, which I don't know why he would do that, right? Then I can extrapolate and know that he's got it. So that's one way I explain. I don't think the two are in juxtaposition at all. Not to mention that most famous scientists, Mendel, right, Newton, et cetera, were all very firm believers and they didn't see any contradiction at all. There's a lot of answers to that question, but that's one answer I give. So you can feel free to use that one if you want, okay? You probably don't get asked that a lot here, but you will in what I like to call the real world, okay? Where if you haven't noticed, most people don't follow Jesus. At least certainly not in the way that we do.
You get concerned questions about evolution, how that might fit with your faith.
The E word, yeah. That's the second most common one. Well, what about evolution, right? Which is an even, I think, an even sometimes more complicated answer. But there's different ways to answer that within Christian orthodoxy. But mainly my answer goes to the difference between science and philosophy. Right? Belief in God is a worldview, right? It's philosophy. Science certainly is related to it, but it's not. Right? I can't measure angels in a spectrophotometer. It's not science. Science doesn't answer the question of why are we here. Right? It doesn't answer whys. It doesn't answer morality. It doesn't answer lots of questions, right? That's why you have a worldview. So part of creation is worldview. Why are we here? Who put us here? Okay? Whereas evolution is a scientific theory. What I'm trying to say is the idea that if evolution is true, then there's no God, there's no logic in that statement at all. Science, evolution can't prove God's not here, God wasn't involved. It doesn't even ask that question. It's science. Science only looks at the natural world. Science never looks at angels and God and cause, right? Did God want to do that? Science just goes, I don't know. Right? So my question is, evolution versus creation, which is you hear all the time, is like apples versus oranges. You're comparing different things. Right?
Before we open all those topics to different things, you can pick them up over at lunch. We'll be at the Corner Cafe in the Joyce Room. If you want to pick up more on this conversation, go out there. Thank our speaker one more time.